This is a repeat recording for protein synthesis, as I've managed to fail to record it in both class sessions. So this is being done away from a teaching situation, and therefore I hope I cover everything that I need to. You need to be familiar with the section on nucleic acids, so you need to recap that first. And you need to refer to your notes handouts, and there are several sheets in there that refer to protein synthesis. So I will try and refer to those appropriately. That's not handbook, sorry, that's handouts as we go along. Okay. Now, I'm going to start in a slightly different place from where I was when I was teaching this over the last two sessions, and start by talking about the nature of the information held in DNA. DNA, if you remember, is a double-stranded molecule. It's two polynucleotide chains. One of these is described as the template strand. The other is described as the non-template strand, and that's also sometimes known as the coding strand, which, yes, is a mistake I've been making for years and calling the template strand the coding strand, so I don't now use that word unless I have to. The template strand might carry a sequence of bases, such as, it doesn't matter what the sequence is, but I should just put in a random selection of the four letters that we can use. And of course the non-template strand will carry complementary bases. So where there's an A on the template, we'll have T on the non-template. G and C are complementary to each other, and the rest of it would look like that. Now, the function of the non-template strand isn't clear in protein synthesis. The non-template strand is important when we get round to cell division and needing to replicate the DNA to make a copy of it so that each new cell in the division process can get a copy of the original cell's DNA. But at this point, the non-template strand is not important. The template strand is what's carrying the gene, if you like, or genes, about 1,000 on average per chromosome. And the gene, therefore, is a sequence of bases that are going to code for the amino acids which are joined together to make a polypeptide. So you might also need to recap, apart from nucleic acids, I would suggest perhaps recapping protein structure. A protein is a polymer, it's a polypeptide, and is formed from, well, anything from 2 to several thousand amino acids joined together in a chain, and that's the sequence with which those amino acids are joined together is absolutely critical to the way that the protein forms, the shape of the protein, what its job is. And if you were to put together those amino acids in a different sequence or put different amino acids in, you wouldn't get the same protein. And that's something that we consider when we move on to the section on mutations. Now, given that there are 20 different amino acids that appear in proteins, we've got to be able to have a code in the DNA that enables us to select one of those at a time and insert it into the growing polypeptide when we get to that point. Given, however, that there are only 4 bases in DNA, we cannot use one base to code for one amino acid. Otherwise, we'd only be able to code for 4 amino acids. And clearly, if we've got 20 different ones, we need more than that. We could, however, read this template strand 2 bases at a time. But if we did that, we would only get 16 combinations, which I will go through them. It's AA, AT, AG, AC, TA, TT, TG, TC, GA, GT, GG, GC, and CA, CT, CG, CC. So you've only got 16 options if you read it 2 at a time. So we have to move up another step, and the smallest number of bases that we can read at a time to give us at least 20 combinations is 3. And that is how it works. So we have a group of 3 bases in the DNA code is the minimum number that we can have to code for a single amino acid. Now, there are 64 possible threesomes, or what we would call triplets, and those 64 therefore provide us with more than enough code for the 20 amino acids that we need. And in fact, the spare capacity is actually quite useful. All right, so a gene in DNA then is made up of a whole lot of triplets. A gene will also contain some non-coding DNA, but we'll come back to that when we start looking at the actual process. This code, before we move on to the process, the code is described in 3 ways. And the terms that we use, the code is described as, first of all, we describe it as being universal because all living things have the same DNA code for the same amino acids. So it does at least show that living things are related to each other, that this code is the same no matter what organism you're looking at. The second part of the description is that it's non-overlapping. And if you look at the diagram here, what that means is that the bases in the triplet that I've identified there, AAG, do not form part of the next triplet. The next triplet is CTA. So no base in any given triplet forms part of another triplet. Now, the use of that is that if we get an error in the DNA, if we get a mutation, if we get damage, and let's say, for example, that the G in that first triplet was replaced by another letter, it would only affect that one triplet and therefore would only affect one amino acid. It wouldn't affect the next one. So it does build in a little bit of safety because the effect on one amino acid might not be significant enough to cause problems. So the non-overlapping code means that bases only form part of one triplet. The third descriptive word for this code is that it's degenerate. Now, degenerate sounds as though it's, I've spelt it wrong for a start. Let's try that again. Degenerate. Degenerate. The opposite of degenerate is pure, and a pure code would be one triplet, one amino acid. All right, but given that we've got 64 possible triplets and we've got 20 amino acids, it means that a lot of the amino acids, in fact most of them, have more than one triplet code. And this also then means, rather like the overlapping description, that if you get an error or you get a change in one of the triplets, it might actually still code for the same amino acid. So it might not cause a problem. So the fact that it's non-overlapping and degenerate does provide a certain amount of safety in that if you get a mutation or damage to the DNA that changes a triplet, then it will only affect that triplet, that's the non-overlapping bit, and it may still code for the same amino acid, that's the degenerate bit. Now we've got our code, we can start to think about how we're going to decode it and use it to build a protein. So we can look at the actual protein synthesis process. And we have 3 stages to this. Now, they don't actually occur sequentially, one after the other, but, well, transcription is the first part of it. But the one that's usually written second, activation, is going on all the time, so it doesn't happen after transcription. That's just a continuous process. And then the third part of it is translation. Now, I'm going to put at the side of these just a very, very simple description of what's going on here. Transcription is the first part where we've got to be able to copy out the instructions that are in the DNA. And the DNA triplets are transcribed to give messenger RNA, and we met that in the nucleic acid section. And the messenger RNA groups of 3 bases are not called triplets, they're called codons. So every triplet in DNA is transcribed using a base pairing process to produce messenger RNA codons. And we'll look at each of these in more detail shortly. The problem with DNA is that it's like a reference library. We've got one copy of it and it sits in the nucleus. It doesn't come out. And transcription is a process of going into the nucleus, opening up the DNA, copying out the instructions we want as many times as we think we might want them, and then taking those out of the nucleus, closing the DNA up again, and using the copied out version, the messenger RNA, for actually making the proteins. And you know, the number of copies that you can make, it could be a few tens, hundreds, thousands. It depends on what protein's being produced. Something has to trigger that process, and this is one of the things that we come on to in the final biology unit when we're talking about control of gene expression. But to give you an example, if, just going back to the last unit, the physiology unit, when we were talking about controlling blood glucose levels, if our blood glucose rises, we need to produce the hormone insulin. Now, insulin is a protein. There is a gene which gives the code for making insulin. And so the production of insulin involves the activation of that gene or the expression of that gene. And it's the rise in blood glucose level that will then cause some sort of control process to open up the DNA at the point where the insulin gene is. We then make as many copies of the insulin gene as seems to be useful. That will then leave the nucleus, and it's that messenger RNA template, well, not template, but messenger RNA set of instructions that will go out into the cell and be used to build the insulin. Okay, activation. This is a process that's going on all the time in the cytoplasm. And this is where our transfer RNA combines with amino acids depending on the sequence of bases in part of the transfer RNA called an anticodon. But I'll fill the details in for this in a bit. So transfer RNA picks up amino acids. So it is transferring, that is its job, hence its name. And then the last stage, translation, is where the messenger RNA sequence, the codons if you like, let's put codon sequence, is turned into a sequence of amino acids which forms a polypeptide. That happens at ribosomes. If you think back right to the very first unit that we did, the cell biology one, ribosomes, particularly on the rough endoplasmic reticulum, will receive the messenger RNA, they'll receive amino acids carried by transfer RNA, and they'll convert the messenger RNA codon sequence into a sequence of amino acids. All right, if I put that sequence of amino acids, and that is what we call our polypeptide. So we'll have a look at these 3 stages. We don't really do very much on the second stage, but the first and the third we can look at in somewhat more detail. And you'll find in your handouts that you've got a sheet that's headed up Information Coding and Protein Synthesis. The first part of that tells you about the code being universal, degenerate, non-overlapping, and all of that. And then down at the bottom of that page, it goes on to protein synthesis. It lists these 3 stages. And then just at the bottom there, it starts with transcription. And the first thing that has to happen is that the 2 strands of DNA have to be separated. And they're held together by hydrogen bonds, if you think back to the structural bits that we did. And those hydrogen bonds have to be broken and the two strands separated at the area where we've got the gene that needs to be expressed. We then have the formation of messenger RNA, but it is in a form that has to be modified. So I'm going to produce a diagram which will fit into the space over the page in your handouts. But if we just have a look at what I've written there, hydrogen bonds holding together the two strands of DNA are broken in the region to be copied. That's the gene to be expressed. The DNA double helix unwinds. The code on the template strand is transcribed into a single strand of pre-messenger RNA, giving a sequence which is complementary to that of the DNA. Now, it's complementary in the sense that you've got the base pairing like you have in DNA, where it would be in DNA, it would be A pairs with T and C with G. But when we're forming messenger RNA, we can't use T because RNA doesn't contain thymine, it contains uracil instead. So we have to remember that as we're going through. So we'll start with that part, and then I'll move on to the other part of the description when we've done the first bit. And it's easiest to do this diagrammatically. So if this is a piece of DNA that's been opened up at one part, let's call the top strand the template and the lower one the non-template. And I'm just going to put a sequence of letters A, T, G, C, A, T, G, C, A, T, G, C, odd A at the end. And then the complementary DNA bases would be on the other strand. And where it's been opened up, this is only a very small part I've opened up on here. So this is probably only part of a gene, and it would keep opening up. It doesn't matter which way this is going, left to right or right to left. It will actually, in terms of which way the enzymes can work along a molecule, if you think back to the nucleic acids bit, whether it's 3' to 5' or 5' to 3', and I'll show you a diagram of that on a PowerPoint when I've done all this, that'll make that clear. But as far as we're concerned, it doesn't matter too much. So let's say it's going, oh, I don't know, I'm going to start at the left-hand end, and we've got an enzyme involved here called RNA polymerase, and RNA polymerase builds the polymer. The fact that it says A's on the end means it's an enzyme, so it's just building the RNA polymer, and it brings in nucleotides to match or to complement the nucleotides on the template strand. So here, for example, it brings in a nucleotide with cytosine. We move along, it brings in a nucleotide with guanine. We move along, and it brings in a nucleotide not with thymine, because this is messenger RNA, it's got to be uracil. So we would keep going like this, and then the next one to come in, all right, this would be the next nucleotide to be joined on, and then there'd be another one like that going to be joined on. So they're just waiting to be added to this growing chain that we've got here. So this is our pre-messenger RNA. And that's got to be processed before it can be used. Now, up in the summary that I did up here, I said DNA triplets are processed to produce messenger RNA codons. So, if this is a triplet, if that's a sequence of bases that's coding for one amino acid in the DNA, then the complementary section of the messenger RNA, that is what we call a codon. And very often, if you're looking at reference material for the sequence of a piece of DNA, or for producing a polypeptide, it'll usually be the messenger RNA codons that are used to tell you which ones come next. Now, this idea of pre-messenger RNA is due to the fact that Genes contain coding and non-coding DNA. So we've got two parts, all right? So it's a section of DNA which consists of the coding bits that are actually useful. So we've got coding sections that actually say which amino acids are going to need to be put together. So you've got the coding sections. We refer to those as exons. X, the EX there meaning executable. And we then have non-coding sections. Now, the problem with this non-coding idea is that it makes it sound like they don't do anything. But in fact, the non-coding bits, we used to call it junk DNA, do now seem to have some function in producing little bits of RNA that help to control gene expression. They're called introns. We also get non-coding DNA between genes. But the problem is here that when we've transcribed the DNA code into pre-mRNA, it's pre-mRNA because it's still got all the introns in it. It's got the bits that actually aren't useful for coding an amino acid sequence. You could think of them a bit like adverts in a television program, where you get a bit of program, then an advert, a bit more program, then an advert, a bit more program, then an advert, and then a bit more program. And if you were to record that, you'd probably sort of cut the adverts out when you were watching it back. And we do the same sort of thing in protein synthesis, that the pre-mRNA is a molecule which contains complementary copies of both the exons and introns. So let's say we've got an exon first, then a little intron, then another exon, then a bigger intron, then another exon, then another intron, and another exon at the end. So we've got 4 useful bits. Let's say these here are our exons. They're the useful parts that we want. But we also have 1, 2, 3 introns. And the introns have to be cut out, and then the exons have to be joined back together again. And the term that we use for this is that they are spliced out. Splicing is a term that actually is often used for joining pieces of rope together so that you can remove damaged parts and join the good bits back together again. But here we splice these out, and that's not then to say that they aren't any use, because it would be a bit wasteful of the body to keep producing these, to put resources into making things that weren't actually any use. We used to think that they were just bits of code that were left from evolution and perhaps from viruses getting into our DNA and that sort of thing. But we now do know that a lot of these little short pieces of RNA do actually have roles, and I keep promising I've got at least 1, possibly 2 books I need to read before we get on to the final biology unit, so I can tell you a little bit more about how these little bits of RNA that are spliced out might have a role in controlling gene expression. So what we're left with here is the remaining parts are joined back together again, the exons, and so we now have a smaller piece of functional, all right, these are the exons, and we have a smaller piece of functional messenger RNA. That leaves the nucleus, goes to a ribosome, and is used in protein synthesis. So this leaves the nucleus. Okay, so the process of transcription is not just copying the DNA code into messenger RNA, it's copying it into a form that then has to be processed. We might call this post-transcriptional processing. The next stage then is going to be activation. Now, activation is going on all the time. This is a continuous process in the cytoplasm. And it really just relies on the transfer RNA molecules. And we had drawn them, or I think we'd seen them on the PowerPoint, as having what's often described as a clover leaf shape, sort of like that. They bind with amino acids, right, at that point. But which of the 20 amino acids attaches is determined by a sequence of bases down here. Okay, so that could be AAA, ACG, CGU, whatever. But that is what we call the anticodon. We very often use a much abbreviated version of this molecule when we're trying to draw the process. So very often it'll just be something like this, which is how I'm going to do it. All right, so the amino acid would be there and the anticodon there. All right, so a much simpler version. But this is the equivalent of going off to get the ingredients for a recipe. The messenger RNA is the recipe. And so our final step in all of this is going to be reading the recipe, putting the ingredients together, and making our product, which in this case, of course, is a polypeptide. So our final stage then, and the sort of most significant part of this, is creating our, it's not a protein at this point, it is a polypeptide. It's called translation, and it translates the messenger RNA sequence into a sequence of amino acids that we call a polypeptide. And I'm going to do this largely diagrammatically. And the processes that occur after translation, so post-translational processes, are not something we'll go into, but obviously once a chain of amino acids has been formed, it can then twist, it can fold, it can join with others. So there is a lot more to it than just producing a string of amino acids. Now, if this is our messenger RNA, it's been processed, so this is now a functional version of it. I need to put a sequence of bases on there that will actually make sense. And so if you just bear with me, I've got a set of codes that I need to use here. We would have a very definite code at the beginning of an RNA sequence for making a polypeptide. It's always the same sequence, it always brings in the same amino acid, and that's methionine. Other than that, it could be any sequence we like, so I'm just going to do something that makes it relatively easy for me to check these off a reference sheet. And there we are. That's a little bit of messenger RNA. Now we've got a codon at the beginning that reads AUG, followed by another one reading UUU, followed by another one reading UGU, followed by another one reading GGA. But of course, this would go on probably for hundreds, possibly thousands of triplets. This could be a very short protein, but I'm just using this to illustrate. What we need attached to this is a ribosome. Ribosomes have two parts to them which sort of fix themselves around the messenger RNA, large subunit and a small subunit. And for the purposes of this, it's moving left to right. So that ribosome has already been to AUG and UUU. It's currently sitting on the UGU codon. Now, the fact that it's sitting on there means that it will bring in a piece of transfer RNA with a very specific amino acid that is determined by the anticodon, which of course is complementary to the codon, and it's going to have the code A, which is complementary to U when we're looking at RNA, C, and another A. Now, UGU as a codon in messenger RNA actually codes for the amino acid cysteine. However, that wasn't the first one on the list. The amino acid that was added when the ribosome was sitting on the UUU bit was phenylalanine. And the one that was added when it was sitting on the AUG bit was methionine. That is always the one you get at the start. The next one that will be added when the ribosome moves to the right and sits on the GGA over here, the next one that will need to come in, so let me sort of put it wonky so that it's not quite sitting on there yet. This will have the anticodon CCU, and that will be bringing in the amino acid glycine. And there are tables and charts that actually tell you this. All right, and this is what we mean about the universal code. It will always be the same amino acid brought in for the same codon. So you have codon tables that tell you which messenger RNA codon gives you which amino acid, or sometimes a slightly different version. You have codon wheels. Sometimes they're done as part of the DNA code instead, but it's just a matter of decoding. This thing here then is the ribosome, and the ribosome holds together all the components that are needed for protein synthesis. So we've got the messenger RNA, the transfer RNA, amino acids, and then of course if you're going to join amino acids together to make a polypeptide, you're going to need enzymes to do that. And I'm sure that's not all that's involved there, but you know, it's holding together the components, acting rather like a kitchen. So if the messenger RNA is the recipe, the amino acids are the ingredients being brought in by transfer RNA. The ribosome is the kitchen where everything is put together and the final protein is formed. Sorry, not the final protein, the final polypeptide. So we're building a polypeptide up here. That's what this is going to be. All right, so this is our growing polypeptide, not yet a protein. Whoops, didn't quite leave enough space. The other thing to add here is that if you've got a piece of messenger RNA, then you want to get good value out of it. You know, you don't want to just use it once. And so this could be read hundreds of times, thousands of times. And in fact, from the single gene in a piece of DNA, you could have hundreds or thousands of copies of messenger RNA. In fact, it's easier to find that than it is to find the gene sometimes. And each piece of messenger RNA can be reused, reused, reused. And you can have several ribosomes moving along a piece of messenger RNA at any given time. So if this piece of messenger RNA has got 3 ribosomes moving along it, what you end up with is, well, the one at the far right here has got pretty much a complete polypeptide. All right, so that's got a great string of amino acids there. The one in the middle hasn't gone quite as far. That's read about half of it. And the one at the beginning's, well, maybe thirds and 2/3 there. But the ribosomes are moving left to right there, and the one on the right is almost finished. The one on the left has just about started. And so you get several ribosomes moving along a piece of RNA, you know, decoding it all at the same time. Now, what we've got then is an overall process of protein synthesis, which should match what you've got in your notes, in your handouts there. You've got my own written version of it with gaps in to put the diagrams in and a little summary. You've also got a couple of sheets which I've taken from an A-level biology notes source that's online and is rather good actually. It takes you through transcription, post-transcriptional modification, that's the splicing out of the introns, and the process of translation as well, and yeah, with 6 diagrams rather than just the one that I've put up there. And then it also mentions post-translational modification as well. Following that, there is an exercise for decoding the DNA sequence, or matching up the base triplets and codons and anticodons, which I'm now going to run the PowerPoint that goes with this that you'll find on Moodle. And one of the slides includes this activity. So when we get to that, I will fill in the answers, but it would make sense, you know, obviously this is something you have to learn to do for yourselves. Okay, on to the protein synthesis PowerPoint, which, as I said, you'll find this on Moodle. You haven't got a printed version in your handouts because I think that's probably a bit over the top. I produced this last year when it became evident that I'd been using a wrong piece of terminology for rather a long time. And a lot of this we don't need to spend much time on, but it's quite a useful PowerPoint in terms of giving you definitions. And like I said, I will pause at the activity, and I'll fill in a couple of the examples and leave you to do the rest. Now, this, the first couple of slides in here are about key components. And the reason that this is in red is because this was where last year I had to correct what I'd been doing just as we got to revision, which was a wonderful time to find out I'd been using the wrong word. DNA then, it's two polynucleotide strands, the template strand and the coding strand. And I used to use the word coding to mean the template strand. So I would prefer to use template and non-template. The word coding actually is related to the fact that the non-template strand contains the same sequence, of course, as to what you get in messenger RNA, apart from having the thymine in DNA but uracil in RNA. But I think it adds confusion, so I tend to stick with non-template strand now because it's just easier. So you've got an outline there of the DNA, the two strands, template and non-template, the nucleotides that you find in DNA, then a little overview of RNA, but we've had all this in the nucleic acids session anyway, so that's not a problem. A gene that we've now just looked at, a sequence of base triplets that code for amino acid sequence of a polypeptide. The promoter region, we haven't done that yet because that's coming into gene regulation, and that's part of the final unit. It doesn't come into this one. So we will be looking at promoters at some point. RNA polymerase, we've just met that one in terms of making messenger RNA. So most of this we're now familiar with. And then the different sorts of RNA, messenger and transfer RNA and their jobs. And a ribosome reads the messenger RNA sequence. I don't know that it does, and that's another one of those situations. You've got to be a bit careful about, well, the word we would use is anthropomorphizing, giving something that's not a living thing in its own right, sort of human qualities, so a ribosome can't read. But it moves along and decodes the base sequence, so I suppose that is like reading, isn't it? And it holds everything together. Now, this is quite a nice little diagram, just simplifying the whole process. So if you look at the top, you've got something that represents a DNA molecule. A section of the DNA molecule is a gene, and a small part of that is this blue sequence across the middle there, going from 3' to 5'. And that's the template strand, and it shows you that a triplet on a template strand is 3 consecutive bases there. By the process of transcription, we produce the red molecule, which is the messenger RNA, which of course will go from 5' to 3'. If you look back at the structure of DNA, you'll work out why. And a sequence of 3 bases in messenger RNA is a codon, not a triplet now. And then through the process of translation, those codons are then used to put together a sequence of amino acids that makes a protein down at the bottom. So that particular one, we've got, I think that's tryptophan, phenylalanine, glycine, and serine. So I quite like that one as a summary. So worth using if you're finding this quite difficult. Then that is a good diagram to have by your side while you're looking at written descriptions. This is a slightly more elaborate version of the whole process, showing, rather than just using straight versions of DNA, using the helix version, helical version. And one of the reasons I put this in is because it labels the two strands of DNA as the template strand down at the bottom. All right, so this here is the template strand, okay, the one that's actually being used to create the RNA, whereas the one that isn't forming a template up here is known as the coding strand. And you can see that the RNA polymerase, the big sort of lilac-coloured blob in the background, is moving left to right here. The DNA is being unwound, the RNA is being formed against the template strand, and as it moves, then the DNA can be wound up. It's rewound again if you only need one copy, or I suppose it can be done over and over again, can't it? And so transcription then is this production of the RNA from the template strand of the DNA. Now, there is the activity then that you've got in your handout file. The idea with this is to take each of these examples down the bottom, and you will get something like this in the exam, but without the big table at the top. You do not need to know which amino acids have which codes. All right, so you won't need to use a lot of your notes sheets in the exam, because it is open book and you're going to be allowed 2 complete A4 sheets of notes. You don't have to use part of it up with a codon table like that. The idea is just that you are expected to be able to match up the complementary bases. So if we have a look at what we've got here, we've got ATG on the non-template strand of the DNA. Now the complementary basis for that on the complementary basis for that on the template strand, of course, will be, if you remember your base pairing, it's T, A, C, because A pairs with T and C with G. Now, from the template strand, of course, we make a messenger RNA codon through the process of transcription. And again, we use complementary base pairing. T will give us A on the messenger RNA. A would give us T if it was DNA, but it's not. It's RNA, so we get a U. C goes to G. And then moving down the same sort of approach, A would give us T if it was DNA, but it doesn't, so we get U. The U gives us A, and the C from the G. Okay? You don't need to put the lines in there, those are just my talking lines. Now, to work out which amino acid we get from that, you have to look at what we've got up here, and it says messenger RNA codons. So we need this line. And so the base sequence that we need to find is AUG, and you will find that here. Not surprisingly, it's the start codon, and the amino acid that's put at the start of all polypeptides is methionine. I think it gets trimmed off afterwards. So I will put MET, but also start. All right, put a slash. So MET or start. I'll take you through the next one as well. If the messenger RNA codon is ACU, well, let's go back up to start with. Well, A pairs with, if we're going to DNA, it's got to be a T. C will pair with G. U will pair, well, U is the replacement for T, so that will pair with A. And then the non-template strand in the DNA, you get T with A, G with C, A with T. And then for the transfer RNA, we'd be looking for A pairing with T if it was DNA, but U because it's RNA, C with G, and then U with A because U is the replacement for T. And so we are looking for that amino acid. All right, this row is the one that gives us the code on the table up there, ACU, which is here. So our amino acid is threonine. And the rest you do the same way. It's just important to remember A to T, G to C when you're pairing the two DNA strands. A to U, U to A, but G to C if you're pairing RNAs. But then you've got to be careful in that transition between DNA and RNA. All right, you still get A, C, and G, but T and U perform the equivalent pairing. All right, so they both pair with A. All right, so yeah, give that a go. Get used to that, yes, expect to see something like that in the exam, but without the final row on the table and without the messenger RNA codon table. Okay, an overview of what's happening then. We have our original gene has this area that we'd call a promoter. That's where you've got your on switch, if you like, that triggers the transcription process. And the gene itself consisting of introns and exons. The transcription process produces this pre-mRNA, sometimes called nuclear RNA because it exists in the nucleus and stays there. That doesn't really help. But the pre-mRNA has to be processed by this splicing that removes all those red bits, the introns, leaves the blue bits to get stuck together, the exons, and then those can be translated to give us an amino acid chain, our polypeptide. And then you can have post-translational modification, which involves the twisting and folding to give you the final structure of the protein. As an overview, that's quite a good diagram. Starting with DNA in the nucleus, the DNA opens up. All right, so it's have a look at that. So this is your DNA when it's not being used as such. Here's the DNA being opened up. Transcription is taking place and producing the RNA. The RNA then leaves the nucleus, goes out to the cytoplasm, and is picked up by a ribosome, which we could label on here. So there's our ribosome down there. And the ribosome moves along the messenger RNA from the 5' end to the 3' end, bringing in transfer RNA molecules that have complementary base sequences to the codons on the messenger RNA and carrying appropriate amino acids, which are then joined together so that we get our polypeptide, which then becomes the protein. Please don't get confused between the sequence of bases and the chain of amino acids. Each amino acid requires 3 bases in the nucleic acid to code for its insertion into the polypeptide. But it's very common for people to get confused between base sequences and amino acid sequences. So this is something that you do need to put some effort into because it will be part of the exam. You will be expected to know which stages do what. You will be expected to be able to decode some base sequences and, or not decode them, but to be able to work out what the complementary bases would be. Okay, that I think is the end of the protein synthesis section. Make sure that you refer to your handout notes and to that particular PowerPoint on Moodle, and hopefully you'll be able to look at some of the exam questions in the handout folder as well. And this particular session is then followed by the one on mutations. The significance of mutations, hopefully you can see that from this, because if you change a base sequence, of course, you can see you're going to get a different amino acid brought in, you're going to get a difference in the amino acid sequence of a protein, so it won't necessarily be the same protein. But there are a lot of potential types of mutation that we'll be looking at.